Okay, so now let's start. My name is Yiming Chen, and I'm from Origin Lab Technical Support Team. And I'm very glad that I'm going to share with you today's webinar topic, Advanced Statistics. So before the webinar, again, if you have any questions in the middle of the webinar, please type it in the Q&A panel. My colleague Zhen Xiao will help to answer it. And towards the end of this webinar, we will still have a polling. So please leave your feedback and suggestions. We really appreciate it. Okay, so before we start to the detailed topics, I'm going first to let you know some of the resources in Origin that where you can learn the statistics features. Okay, so first is the Learning Center. If we go to the Help menu and go to Learning Center, or you can click F11 as a hotkey, and you open this like window. On the left, you select the analysis samples. And here in the drop-down, you can select from various different statistics tools in Origin. So each one will have a project which has the data and the analysis. It has detailed explanation of the analysis and the explanation of the data and the result. So just feel free to try each tools. And also we have very nice documentations. If you go to I just Google Origin Lab tutorial statistics, you will come to this statistics tutorial page where you can also find the detailed instructions of each tool. Okay. Again, don't forget to send us emails. You can contact our technical support team, and we are happy to answer any of your questions regarding statistics. Okay, so in Origin, we have this statistical menu where you can select from different tools. But just to let you know, in order to see the menu statistics, you have to activate a worksheet, because the menu item is related to the windows that you are open. So statistics menu are only available for workbook window. So when you click, you will see a bunch of statistics tools of different categories. So in this webinar, I'm going to cover a couple of advanced statistics tools like ANOVA, Non-parametric tests, survival analysis, and several multivariate analysis tools. Okay, and we also have this app panel on the right side of Origin. You may already have known. So we have over 200 apps and like more than 30 statistic apps. So to find a statistic apps, you can simply click on the Add Apps button. It will open this App Center window, and you can simply type statistics. And it will list all the statistic apps in this window. And simply click and download, and it will show on this app panel. Click the each app to launch the app and run particular tool. Okay. Okay, now let's go to our topics. So first, I'm going to talk about one-way ANOVA. So yeah, so ANOVA is kind of a tool to analyze the grouped data and to compare the group means among multiple groups. So here we have this data. It's like the nitrogen absorption for different plants. So the column B is the measurements, and the column A is the group, has the group info. It has in this column we have 4 plant types. Okay, so this kind of a structure of data is what we call the index data. So which means that you have one column for the measurements, another column of the group info. So in Origin, we have a different data organization structure. So which is called the raw data. It has, so in this case, each plant has its own column. Okay, most of the statistic tools involving the grouping thing it supports these two different data structure. Okay, so now let's do a one-way ANOVA. So we want to compare the mean of the 4 groups. So as you know that in order to do one-way ANOVA, we need to, the data itself has to satisfy some certain requirement. Okay, it has to be normally distributed for the population of each group. So in origin, we have the normal test tool to test whether the data is normally distributed. So if we go to statistics, let's go with the index data. Okay, let's go to statistics and descriptive statistics, and we find this normal test tool. And here we can select the data range as the column B is our data range, and the group is column A. Okay, then we can choose different normal test method in this tab and click OK. Okay, so if we go to this result sheet, in the blue footnote, it gives us the conclusion. Okay, so yeah, it just tells that the population follows the normal distribution. Okay, so this is the assumption has to be fulfilled for one-way ANOVA. Okay, so once we know that the data is following normal distribution, then we can run the analysis. Okay, so let's go to ANOVA, one-way ANOVA. So in this case, I'm using the raw data instead of the index data. So as I said, both data are supported by the by this tool. And in this case, I have 4 levels, 1 factor, but 4 levels for that factor. And here we can click this button, and it says select from the worksheet. I simply just highlight the entire worksheet of the raw data and click OK. Then it will, it's automatically fill each group with the proper column, and also the level name is auto filled. Okay, we have 4 levels. Okay, and we can go to other tabs. For example, we want to check the two-key method. We use the two-key method to do mean comparison, and another thing we can test is equal variance. It's another assumption that has to be fulfilled. Okay, the power analysis. So based on this sample size, we want to know the actual power. Okay, and for the output, we there's a different settings for the output, and we want to check the bar chart, box chart, and the mean comparison plot. Okay, so before I click OK, I will just let you know that in Origin you can save the settings of the dialog. So here's this button, and you can save this as a theme. Okay, let's say my test. So you can save it to a theme. So next time when you open this dialog again, you can simply load the theme to load all the settings. This is a very convenient that you want to repeat the analysis from data to data. Okay, and another thing I want to mention is this related apps button. If you click this, it will open the App Center window. It will list all the related apps to one-way ANOVA. So yeah, this is for one-way ANOVA. It only lists the paired comparison plot. Yeah, this is a nice app. My colleague Isha introduced this morning. So for different tool, it will list different relevant apps. Okay, you can choose from. Okay, once we are done and click OK. If we go to the result sheet, and let's see what what result we can get from this test. Okay, so first thing we can test check is the overall ANOVA. So the p-value is less than 0.05, the significance level we set, which tells us that at least one group, its mean is very different. Okay, you can also check the footnote for the conclusion, and you can also check the statistics. And under the mean comparison, we check the result for Tukey test, and especially to note this significance mark. So 0 means there's no significant difference of mean, and 1 the difference is significant. So we can tell that plant 4 to plant 1, plant 4 to plant 3, plant 4 to plant 2. So they are the main difference are significant. Okay. Also, the homogeneity test of the variance. So it also tells us the variance are all the same in each group, which is nice because this assumption has to be fulfilled. And the power. Okay, so under the 0.05 alpha, the significance level, we get a very high power. So the type 2 error is very small. Okay, and we can also check some plots. So the box chart. Yeah, we can tell that the plant 4 has a little bit lower mean than the other 3 plants. Okay, but to tell whether they have a much difference statistically, we have to go check the analysis table. So under the mean comparison plot, sorry, let's see. Okay, it's here. We can see this vertical line at 0. So the mean difference of the other 3 plants across this 0 line means they are not very different. But for the plant 4, the mean difference is significantly different from the other 3 plants. Okay. Okay, so let's go to the other example. One-way repeated ANOVA. So this is a different measurement that, in this case, each subject is multiple different measurements are applied to single subject. So we want to test whether the difference between the mean. So if we do repeated ANOVA test, the assumption for the normal like one-way ANOVA is violated because the measurement of different, in this example, different measurement on the same subject, they are not independent with each other. So so we have to use the repeated one-way ANOVA. So in this case, other than the group info and the measurement, we have an extra column which has the index of the subject. Okay. So for each subject, we have a multiple measurement. Okay. So let's go to the statistics and ANOVA, one-way repeated measure ANOVA. Again, in this case, we select the factor as column B, which is the dose. We have dose 1, 2, 3, 3 levels for the factor, and the measurement is column C, and the subject column is column A. So in this case, we have some missing data in the worksheet. So which is why here is this warning message that tells us that the subject with the missing value will be removed from the analysis. So in the mean comparison, we can still check Tukey. Okay, so yeah, this is just options to show different plots. Okay. Yeah, again, it gives the result sheet, which is similar to the one-way ANOVA. It gives us the Maltese test of the spherical. This is also a test of whether the assumption for repeated ANOVA is satisfied. Okay, and it also gives us the within and between effects table, and you can check. Okay, this is the one-way repeated ANOVA. Let's move to our next example, two-way ANOVA. So in this case, we have this data of systolic blood pressure as a measurement, and we want to check whether the sex and the diet type has effect on the blood pressure. So on sex, we have 2 levels, male and female. And for diet, we have 3 levels, strict vegetarian, lacto-vegetarian, and a standard American diet. Okay. Besides the effect of each factor, we also want to detect whether there is interaction of the 2 factors. So this time we'll use two-way ANOVA. So let's just highlight the measurements column C, and then go to statistics, ANOVA, two-way ANOVA. Okay, so in this case, we are using like indexed data. So we specify the factor column. Column A is sex, column B is diet. And we check the box of interaction, which we want to check. And yes, we can again use the Tukey method and the power. We just to calculate the actual power and the mean plot. Yeah, we can just check the interaction. Okay. Let's click OK. So if we go first to the ANOVA table, and we can tell that for the like individual factor, sex and diet, the p-value is very small, below 0.05. So the sex and diet factor is significant, but the interaction p-value is above 0.05. So at the significance level of 0.05, the interaction is not very like crucial. So this interaction can be removed to from our model. Okay. So for the mean comparison, again, we can check the we can just go particularly check the interaction. So it tells us the significance mark tells us whether the difference is very different. Okay, and also the actual power of this test. Okay. So if you don't want to check the details of this like these tables, you may want just to check the just want to compare the mean. You can use this app, paired comparison, which Ishwar, my colleague, has introduced this morning. So you can simply put the index in the factor column as the group columns and data. And yeah, we just want to maybe show the just a significance mark. And also we use the Tukey method. Yeah, this gives you a nice graph. You will see within the female group and the strict vegetarian, and the American diet has a significant difference, and the lacto vegetarian and the standard American diet has a significant difference. So which is agree with this ANOVA table. See here. Okay. Okay, now let's go to next example, non-parametric tests. So sometimes the assumption to do one-way ANOVA and two-way ANOVA is violated. For example, the measurements within one group is not normally distributed, or the sample size is too small. In that case, running a normal ANOVA is not proper. So in these cases, we will use the non-parametric method, analysis method. So we have this nice tutorial page. If I just bring this up. Okay, so if you check this page, it has the comparison of non-parametric tests versus parametric tests. So with that said, if the requirement for the normal ANOVA test is violated, then you can go to this counterpart of non-parametric tests. For example, in this case, we are going to test the mean of two groups, see if they are significantly different, but the normal t-test, it doesn't work in this case. So we will go to the corresponding non-parametric method, man-Whitney. Okay. So let's just do that for this data. Okay. So we have two groups, A and B, and the aberration is measured in the column B. So let me go to statistics, non-parametric tests, and man with me. Open dialog. Okay. So for group range, I select the column A, the data range, the column B. And yeah, again, we are looking at the 0.05 significance level. Okay, let's just click OK. Yeah, let me enlarge the window. Okay. Okay, so for non-parametric test, it's not using the numerical value of its group. It instead uses the ranking of its measurement to build the test statistics. So in this case, the statistics is the value is above 0.05, which gives us the assumption or give us the conclusion that the two distributions are not significantly different. Okay. So again, when you are not very sure which statistics tool you are going to use, you can resort to the statistics advisor. Okay, so just to show you that in this case, how we can make the right choice. So in this case, we are comparing the mean of two groups. So we are just answer the couple of questions. Compare mean. This is our goal, and we are going to compare means. And I will say they are normally distributed. No, they are not normally distributed. How many groups? We will say one group with two levels, and which subset contain two treatments, paired or not. We are not doing paired, so we click no. Then it will give me this man-Whitney test, and click it, and you can click the open dialog just to run the tool. Okay, this is very helpful to help you to make the decision. Now let's go to next example, survival analysis. So survival analysis is to analyze the time until a certain event is occurred. So for example, like the death of a subject or a failure of a manufactured part. So in this case, we have this data. We have different mice who already caught a cancer, and we applied 2 drugs onto the mouse and see how long they will survive. Okay, so in this case, we have this column C is the factor column. We have 2 drugs, and the column A is the hours the mouse will survive after they take the drug. And the column B is the what we call the status. So 1 means this the mouse is dead because of the cancer. 0 means the mouse is dead not because of the cancer. And 2 means the mouse is survival through the whole process time, the test time. So in this case, 0 and 2 are referred as censored status, which means this subject will be dropped from the analysis. Okay, because they are not what we want to detect. Okay, let's go to the okay. In Origin, we have if you go check the survival analysis menu, and we have 3 survival analysis tool. The non-parametric tool, Kaplan-Meier estimator, the semi-parametric way method, Cox model estimator, and the parametric method, Weibull fit. So in this case, I'm going to use Kaplan-Meier estimator. Okay, in this dialog, I will choose the time range as column A, the number of hours, the mouse survived, and the sensor is column B, and the grouping is column C. Here I'm going to censor 0 and 2. So as I said, if the status is 0 or 2, these subjects will be dropped from the analysis. So for the survival table, we can check the mean estimate, like the mean survival duration we want to check, and the survival plot. We can check a hazard plot, and the equality test. We can check these boxes to test whether the two drugs have the same effect or not. Okay, let's click OK. Yeah, there's some result we can check, like the mean of the survival time, so like 32 hours for the first drug and 46 hours for the second drug. And we can check the survival function, which gives us the survival probability as a function of time. And just apparently the two drugs behave very differently. Okay. And it also gives us the status test with whether the two drugs are very different in terms of curing cancer. Okay, so the conclusion says there's a significant difference. Okay, so this is a survival analysis tool in Origin. Let me go to the next example, clustering. Okay, so we have this big worksheet. This contains the monthly temperature of different months for different cities in US. Okay, so the clustering analysis is just to cluster either the variables or features into groups, or cluster the cities into small groups. Okay, so based on the temperature, we can say, okay, some of the months are like high. We can say it's a high temperature months. Some are like low temperature months. Like we can just divide them into small groups. Okay, let me first go to, we can select statistics, multivariate analysis. And we have two cluster methods. So k-means and hierarchical cluster analysis. I'm using this one for this time. Okay, so for the variables, let me choose from the 12 months temperature. So from January all the way to December. And for the observation, I will choose column A, city. And in the settings, okay, so I will go to the variables first. I want to cluster the features. Okay, so there are different cluster methods you can choose from. And go to quantity. Okay, just leave it as it is. So for the plot, make sure we are going to use the vertical as the dendrogram. Okay. Let me click OK. Oh, there's something I didn't choose. Let me, if I want to change some settings, I can click the green lock and change parameters. So in this case, I'm going to change this, the number of clusters. I want to divide them into 5 groups, for example. Click OK. And let me bring up the dendrogram. Okay, we'll see how the months are grouped. Like the first 3, January, February, March are cold months. November, December, the second coldest months, and the other pleasant months, and the hot months. Okay, and we can also check this dendrogram in different way. Click this green lock, change parameters, and go to plot. We have a different way to visualize the dendrogram. Let me select circular. Yeah, this is just a different way to show this grouping info. Okay. Okay, now this time I'm going to cluster cities, put cities into different groups, but based on the temperature. Let me again go to statistics. Again, let me choose this all the variables. And observation, let me choose the city. And the settings. So in this time, I'm going to cluster observations. Okay. And again, I will choose 5 groups. Let me click OK. Okay, so this is a very complicated tree. Dendrogram shows the grouping of the cities. Okay, it doesn't take gives us too much information, but we can come here to check the cluster membership. Okay, let me visualize this data in a fancy way. So I first copy this membership and put it on the side, add a new column and put it on into a new column. Make sure it's numeric. Okay. Then, because I have this longitude and latitude of each city, I can plot it out as a scatter plot. And this is more or less like the US states map. Then I can use this Google Map tool to add a map beneath this scatter plot. Okay. So when I launch this app, then I simply click this button, set from active graph layer. Yeah, it will use the longitude and the latitude values in the graph to locate the position. Okay, you can let me choose the satellite. Okay, once done, I'm click import as update. Okay, so you will see the map is being put beneath the data. Okay, I can modify the data points a little bit, make it smaller, and give it a different shape. And I can also map the data to its membership. Remember the membership column I put to the right side of the worksheet. Okay. So this shows like in the lower latitude area, the temperature are kind of higher, and at the higher latitude, the temperature is low. So these cities are grouped according to the temperature. If I hover my mouse to this data point, it gives us the information of each point. I can right-click on this tooltip and select preference. I can add more information like the city name. I choose a city, and I can also add the annual temperature to the tooltip. So this is a way to format your tooltip. Okay. Then when I hover over the point, it gives more information, the city and the annual temperature. So there's one special point here. So it's like the temperature is kind of low among all high temperature cities. Okay, so obviously it's in a valley. The city is in a valley. Okay, so this is just to show you the clustering analyst tool. Let's go to next example, PCA. PCA is the principal component analysis. So this analysis method is used to reduce the number of features of the data. So many times, so when you have a large data set with multiple variables, so you want to reduce the number of variables to describe the data more clearly. So in this case, I was first to start with a smaller data sheet and then go to a more complicated one. So this is the data of the food consumption for countries in UK, and each row is a different food, and we want to check how these 4 countries are associated with each other in terms of their food structure. Okay, we can use this principal component analysis tool. So let me first launch this tool, statistics, multivariate analysis. principal component analysis. Okay. So for variables, I will select all the columns. And for observation label, I will select column A. Okay. So in the settings, yeah, I will just extract two principal components. So I select two here. Other quantities you can select, choose from. And in the plot, I will make sure that we will plot the loading plot and the score plot. Okay, let me click OK. Okay, so here is the result. Let's see what information we can get. So if we want to get like the information from this PCA method, first thing we can look at is this eigenvalues. So eigenvalues tells us how much. So we are now extracting 4 principal components, and we can tell like how much variation of the data can be explained by each principal component. So here the first principal component explained 97% of the variation of data. So that, but the second one only adds up to 99. So which tells us that we can just use one principal components to explain the data. That's enough. Okay. So also from the script plot, we can also try to find the elbow plot or elbow point. So in this case, it's at 2, which so tells us one principal component can be used to explain the data. Okay. Then let's go to this biplot graph. Okay, this is a very informative graph. So we see that the North Ireland is separated from the other 3 countries, Scotland, Wales, and England. And we can, if I move this a little bit, we can tell that the North Ireland is very different because it has a much larger consumption of fresh potatoes. Okay, and fresh potato makes a very important effect in distinguishing these 4 countries. Okay, so one simple test we can do is, what if we mask this data? We mask the fresh potato. I use this minute toolbar to mask this row. And if I go back to this plot, you will see this operation lock turns yellow. I can click it and click recalculate. The result will get updated. So if I remove that fresh potato, then the 4 countries are less distinguishable. Okay. So this is one benefit of using principal component analysis. It helps us to understand the data when there are like many correlations among different variables. We can use this method to project the features into a lower dimension and to further check the data. Okay. A second usage of a principal component analysis is that we can reduce the dimension of data. So when people doing machine learning, they have many features. They want to reduce the number of the features so that they can run the machine learning more efficiently. So in this case, I have this data which is linked to a web data source. So if I click the data source, you will see it's linked to this URL. So this is what we use what we call the data connector. Okay, I can click import to import the data from the web. Okay, now I have this data of wine. So the column A is the type of the wines. We have 3 types of wine in column A, and all other columns are the measurements. Okay, so let's see how we can reduce the variables. Let me just select all the columns and go to analysis, statistics, multivariate analysis. Okay. In this case, I'm going to simply go to settings. I can give it a number of components to 4. Okay, so I will extract 4 principal components from the data. Okay, let me just click OK. Again, we can simply check the scree plot. It tells us how many principal components is necessary. If we again to check for the elbow point, maybe there's a big job at this point. So like 1, 2, 3, 4, 5 principal components is enough to explain the data in this case. Okay, so let's go. This time we are going to check the score data. So it gives us the principal components value. Okay, let us go to the original data sheet and let me copy the class label here and paste. Okay, let me print out the PC2 versus PC1 and color that with this label. Okay, so we can see like after doing the principal component analysis, the data can be more separated. You can classify the data with a like linear boundary. Okay, so if I, for example, if I change the PC1 to PC3, so this is definitely not a good option. So PC1 and PC2 is what we are looking for. So once we extract PC1 and PC2, then we can use these 2 components to do further machine learning tasks. Okay. Okay, this is a PCA. Let's go to PCO for Spectrum. This is actually an app requested from our user. So in this case, we have this 20 spectrums. So we have 20 spectrums. Each spectrum is corresponding to one, either it's olive oil or non-olive oil or a mixture oil. So we want to use the PCA to distinguish these spectrum. Okay. So right now I'm showing the data in the column list view. So if you go to view and uncheck the column list view, you can check the data in the normal view. Okay. So column list view is just a different way to view the information of each data column. Okay. So before run the tool, I can check the data using the graph browser tool. If I click plot and go to browser and let's see color line. Okay, so here we can choose a stack line by Y offset. And I can also show the comments, which is the olive level. So if I choose the first 5, we can see the spectrum of non-olive oil, how the spectrum looks like, and I can check other of the olive oil spectrum. Yeah, this is a very simple way to navigate your data. Okay. So now let's run this tool. So this tool is actually from an app. I downloaded from this app, download it into the app panel. So here is the principal component for Spectrum. Let me fire up this app. Okay, so for Spectrum data, I can go to, let's say, select all the columns and leave out the first column. And for the frequency, I will choose the column A, which is the time. And for the spectrum name, I will use the long name. For the group info, I will use the comments. Okay. So again, I can. So this is very similar to the PCA dialog. Other than that, I think, yeah, for the reference spectrum, I will select a different spectrum. Okay, let me click OK. Okay, this gives me this result sheet. So first thing we can check is the loading of the spectrum plot. Okay, it tells us how important the importance of the its data, its time to generate the principal component. So now I use a sample 6 as the reference spectrum. So if we just look at the loading, so for PC1, it has the highest loading. It has the highest contribution at time around 7 and at time around 6 and 7. So which is corresponding to the largest 2 peaks in sample 6. And for PC2, the largest contribution is from the time 4 and roughly 6, okay, which corresponds to these 2 small peak in the reference spectrum, okay. So this tells us how to the categorical to distinguish different spectrums. So if we go to the score plot, and we can see the green dots, which is the olive oil, they are tightly grouped, and the confidence ellipse are kind of small and tight. But for the non-olive oil and mixed oil, they are kind of scattered, not very, not easily to be distinguished. Okay. Okay, now let's go to our next example, the discriminant analysis. Discriminant analysis is a method to do classification. So here I have this classic facial iris data. I have this the species column. I have 3 species, Satosa, versicolor, and Virginica. And for each observation, I have the measurement of its sepal length, sepal width, petal length, and petal width. Okay, so if I simply plot out the sepal width as a function of sepal length, I plot it in the upper graph, and the petal width and the petal length in the lower graph. If we just look at the graph, we can see it's not very distinguishable for the 3 species based on the sepal length and width, but it's more or less, it can be distinguished with the petal length and the petal width measurement. Okay, now I'm going to run this discriminant analysis tool to build a classification model, then use that to do prediction for the new measurements. So first, I'm going to randomize my data, and then I split the data into the training data and the test data. And I will use the training data to build the model and the test data for the prediction. Okay, let me add one column. So to randomize the data, there's a trick that first I just fill this column with some random number. Let's say I will just do normal random numbers. And then I can select this column, go to worksheet, and I will sort the worksheet by this column. Okay. Okay. Now you'll see the data are sort of like randomized. Okay, now let me go to the tool, statistics, multivariate analysis, discriminant analysis, dialog. Okay, so I will use the first 120 rows as the training data and the last 30 rows as the test data. So first of all, I will use the petal length and the petal width. Yeah, because we know petal length and the petal width has a better effect in distinguishing the species. Okay, I will select I just use these 2 features to build the model. And because I only want to the first 120 rows, I can select this button, click the button and select columns. Here in the rows, I click this button and set it to 1 to 120. Okay. Then let me copy it. This is my training data. So the group info is from column E. Actually, this should be selected as the group data. Again, let me select columns and change this to 1 to 120. Okay, I will check this box because I want to do prediction. Okay, let me select the column C and column D. This time I'm going to only need the rows from 121 to 150. Okay. Okay, that's okay. Let me go to settings. So there are other settings like what kind of boundary you want to put. So if I use the linear boundary, I will just check the linear boundary as the separator, and other options that you can choose from. Okay, I will just leave it as it is and click OK. Okay, in the result sheet, what we can see is the classification for the this is for the training data. It gives us the 94.87% for the Rossi color, and yeah, the Setosa is 100% identified properly, and the Virginica has 92%. Okay, and we can also go to the classification summary for the test data, which is let me go to this. Let me go to this test result. This gives me the group of each measurements for the test data. I can just copy this, the predicted value for the group, and I go to the initial data because it's from 121 to 150. I will simply copy here, and I add one column, and I will simply type in the formula compare. I will just compare the predicted value with the original value, column E and column G, and let's see the result. So here, 1 means they are predicted correctly, and 0 means they are not predicted correctly. I can use this data highlighter tool. So for example, I click the data highlighter and I click on this data. It will show this misclassified points on the graph. And we can tell these points are kind of at the boundary of this two groups. Okay. But in general, this prediction tool is kind of did a good job in the classification. Okay. Let me go to next example, the support vector machine classification. This is actually a Python example. So I'm going to show this in our upcoming Origin 2021. So because this use some Python feature in our new version. Okay. So here I have this data, like XY data, and also I have this group info. So when I plot it out and color its data points with this group, I got this graph, and I want to use the support vector machine to classify the data points. So here I have this Python code, and so in Origin to run a Python code is very simple in 2021. So if you are interested, please just contact us. We can send you a beta version to test. So here, if I just go to connectivity and the Python console, it will, connectivity, open the Python file. It will bring me to the code builder. And I simply paste the Python code here and save. I can simply click this button to run the Python file. So this is very simple in Origin 2021 to run a piece of Python code. So in this case, if I bring up this code, so actually the building of the classification model is just simply 2 lines of code in Python. So I can just use Python script in Origin to generate this plot. So this is very useful because we know that Origin has a very powerful graphing feature, so we can combine that with Python to generate some interesting results. Okay. So another thing I'm going to show you is there are some new workbook template which is also in Python. So let me go to the new and the workbook. I'm not sure if you know this the window in Origin that gives you a different workbook template. So in Origin 2021, we add a multiple workbook template in Python. So for example, this has this ARIMA, the time series model, the template, which is coded in Python. If I simply click it and open this window, so we can see that the Python code is embedded in this button. If I double click on this text, and we can put the Python code in this behind this button. Okay, then I can click this button to run this. Oh, it doesn't have this package. So so basically, just to let you know that we improved Python greatly in 2021, and you can get access to these new features or new workbook template in this workbook window. Okay. Okay, I think that's pretty much the I want to share today. So before you leave, please, my colleague Jen, can you open up the polling, and you can give us some suggestions and feedbacks. Again, thank you for attending this webinar. Hopefully, it's helpful for you. One user is asking whether the discriminant classification can be used on more than 2 columns. Yes, of course, it can be work on multiple columns. So in the webinar, I will just show an example of using only 2 columns. Yeah, you can use for more columns. Someone is asking whether there's a machine learning tools in Origin. Yeah, actually, as I said, you can just one way you can do is to go to the app panel. We have a couple of machine learning apps like support vector machine, like logistic regression. You can find there. Another thing you can do is try our Origin 2021 beta version. So in this upcoming new version, we support Python code. So if you have a piece of machine learning Python code, you can definitely implement that in Origin. And if you have some suggestions, we can make a machine learning tool into an app for sure. Yeah, just contact us with your ideas. Okay. Thank you again for attending this webinar, and have a good weekend. Thank you. Bye-bye. I'm going to shut off the window.